Welcome to the final lecture, narrated lecture of the course. Human reproduction. I hope that everyone has their parents' permission slip filled out. No? Okay, take a deep breath. And this is going to be a long one, 67 slides, so enjoy. Human reproduction is what we call sex, and that is when you get genes from one individual, the female, add it to the genes of the other individual, the male, and come up with a unique individual, unless, of course, there's identical twinning going on. So, in order to produce this recombination that we call sex, there has to be a unique process involved known as meiosis, which reduces the chromosome number in half, so-called haploid number. In humans, it's 23. When male and female, when sperm and egg get together and fuse, you can restore the 46 human chromosomes, or the 23 pairs. Biologically, this is called evolution. As evolution is allowing animals, plants, fungi, etc., to produce genetic changes that can make the organism better or worse over time, depending upon different environments and things like that. And so it is because of this sexual recombination of chromosomes from two individuals, usually male and female, that these alterations in genes can provide a benefit to an evolving organism or not. All right, so how do we become males and females? Quite easily. While there are 22 regular or so-called autosomal chromosomes, there are two that are unique. Those are the sex chromosomes, the X and the Y. Random assortment during the process of meiosis takes the male two chromosomes, the X and the Y, and provides it onto different sperm. The females always have an X chromosome because femaleness is XX. Okay. So, gentlemen, it is you that provides the sex of any offspring that you will produce via the sperm. Theoretically, half of your sperm will have the X chromosome that you inherited from your mother, and half will get the Y chromosome that you inherited from your father. Thus, it is the Y chromosome that determines maleness. This SEM, scanning electron micrograph, it merely shows the sizes. The X chromosome in humans is normal size. The Y chromosome is not and has very few genes. One of the few genetic signal mechanisms that converts the embryo, the fetus, from female to male is a gene known as TDF, which stands for testis determining factor. About 5 weeks of age, during embryonic development, fetal development, the gene is activated and puts out a protein that codes for, let's just call it maleness. Since the female lacks this Y chromosome and lacks this gene, the females will continue to be run as female. If you were to disrupt either the production of TDF or the gene that produces it is somehow mutated, despite the fact this fetus will have a X and a Y chromosome, this fetus will become female. In other words, in order for the males to develop, they need to activate maleness genes. This chart again shows you the effects of TDF activation, where it takes the germ cell and other cells, converts them to testis, while without TDF, those same germ cells become ovaries. The production of TDF begins the developmental process of maleness, especially the testis. A few weeks or so after the release of TDF, the testis begins to differentiate into basically two layers, the so-called germ layers, which will become sperm, and those supporting cells that surround it, which are known as Sertoli cells, and other cells that are around the germ cells, which are called Leydig cells. And Leydig cells are the endocrine organ of the testis and will eventually start producing the steroid hormone, the sex hormone, testosterone. In the fetus, Leydig cells begin to produce high levels of testosterone very early on in fetal development, in male fetal development. There are two functions for this high level of testosterone delivery in utero. Number one, the amount of testosterone in the male will stimulate the development of male reproductive organs. The high levels of testosterone are there to block the influence of your mother. Your mom is producing lots and lots of pregnancy hormones, one of which are the estrogens. Estrogens, being steroid hormones, will move into the male fetus in order to block mom's influence on testis development, the estrogens. The male produces heaping amounts of testosterone in utero. Again, maleness in utero occurs because of the large amounts of testosterone, which is occurring throughout pregnancy. It isn't until birth, parturition, that testosterone levels drop to near zero. And then, with the exception of a small blip early on within the first couple of months of life, testosterone levels are very low in young boys. And most of that testosterone, if not all the testosterone, is being produced not by the testis, but that secondary sex hormone, gland in your body known as the adrenal cortex. Sometime around 10, 11, 12, 13 years, that wonderful phase known as puberty hits. The testis starts producing testosterone. We can get even more specific. The Leydig cells begin to produce more testosterone, and the levels stay high pretty much through the life of the male. That was the testes. What about the ovaries? To repeat, without TDF, fetuses will be develop. Female fetuses will develop ovaries. Okay. The ovaries and within the cells will release some amount of fetal estrogens as a local paracrine effect, but the most female embryo fetal influences comes from their mother because of the high levels of estrogens mom is putting out to maintain structures within the 9 months of pregnancy. This chart again just shows you, even though this is the amount of fetal hormone levels, this is showing you the amount of estrogens within the fetus, which is very high until birth. After birth, the estrogen levels in the little girls drop until, again, although they drop, but again, don't go to zero because of the estrogens being pumped out by the adrenal cortex. And this stays low until the hell of puberty. So let's talk about the regulation of reproduction via the sex hormones and the influence of the gonadotropins. Okay. We've seen this graph before, figure before. Again, we're going to talk about two types of gonadotropins. Both are smallish proteins. Follicle stimulating hormone, or FSH, and luteinizing hormones, or LH. Both gonadotropins, as they are called, are controlled by one releasing hormone from the hypothalamus, known as gonadotropin releasing hormone, or GnRH. Both LH and FSH influence the gonads to produce and release sex steroids. The gonads also produce another small protein hormone called inhibin, which also feed back, besides the steroids, the sex hormones, to regulate the production of LH, FSH, and GnRH. And we'll discuss inhibin when we talk about male parts. All right. Again, we've seen this figure before. Again, showing you hormone levels starting from birth, hitting that wonderful phase of puberty, and skyrocketing to the roof, where they stay high levels until in the female until menopause hits, and there's a humongous tremendous drop in estrogen levels. For the males, testosterone levels decline gradually through the end of life. Thus, theoretically, a 95-year-old man can reproduce. I'm sorry I put that image into your head. Okay. So, let's talk about puberty. Wonderful time of your life, I'm sure. Puberty is the increased production and release of the sex hormones, which are going to influence sexual behavior, sexual characteristics, hair growth, etc., etc., etc. So what causes puberty? It seems to be the hypothalamic hormone known as GnRH. That is the key to development of puberty, or the initiation of puberty. And why? It seems that the last nerve cells in your brain to develop are GnRH producing and releasing nerve cells in the hypothalamus. These cells develop at pick an age, 9 years old, 10 years old, 11 years old, 12 years old, depending upon the environment, depending upon genetics. But these cells will begin producing GnRH, which will activate within the anterior pituitary the gonadotropic hormones, which will stimulate the gonads to produce sex hormones. Gonadotropins, FSH, LH, play a lot of roles besides regulation and production of sex hormones. They play a role in spermatogenesis, oogenesis. They help in the gonadal structure. They help with final development of the maturation of sperm or egg. And in the case of females, LH and FSH induce ovulation. We've just gone through the slide showing that the sex hormones, estrogens and testosterone, help regulate FSH and LH production, as well as the production of GnRH from the hypothalamus. Well, I should say testosterone does that. So high levels of testosterone will inhibit GnRH and LH and FSH. Estrogens, however, are a little unique because estrogens appear to have actually a positive feedback effect prior to ovulation during the female menstrual cycle and go back to their negative feedback function post-ovulation. And we'll discuss that when we get into the 28-day human menstrual cycle. Okay. There's another hormone involved in the regulation, that is inhibin. Inhibin is released by the Sertoli cells in the testis and the granulosa cells of the follicle cells. Inhibin works to inhibit just FSH release in the anterior pituitary. But FSH is required to do everything that LH needs to do, so it does inhibit the reproductive cycle to some extent. Finally, the effects of sex hormones, especially testosterone, on FSH, LH, and GnRH production produces an interesting 24-hour concentration amounts of testosterone. And you'll see on the next slide that testosterone is actually produced in pulses. High level of pulses causes inhibition of LH and FSH, so that testosterone release will start dropping. As soon as its release starts dropping, GnRH, LH, and FSH will be released by the various glands, which causes more sex hormone testosterone production. So the testosterone starts going up, and these pulses are very interesting. And you kind of see this in this chart here. In fact, all hormones that undergo negative feedback are all released via pulses. It's just more interesting showing testosterone. So growth hormone, TSH, ACTH, also working by negative feedback, are released in pulses. So why pulses? Well, it appears that this pulsatile release of testosterone prevents adaptation, habituation, or short-term inactivation by the hormone that we see in the nervous system. Right? One of the differences between the nervous system and the endocrine system is hormones have a long-term effect. One of the reasons why hormones like testosterone has a long-term effect is its pulsatile release. Second interesting aspect of this figure here is that testosterone levels increase at night and decrease during the day. This is due to the circadian clock in our heads, which influences the hypothalamus and GnRH release. Thus, as testosterone levels are increased at night, it has its most influences at night, which might explain a lot of at least sexual behaviors by males at night. This pulsatile release also occurs again with human growth hormone and with thyroid hormones, which is why there are these anecdotal reports of young males during puberty growing like an inch during the night. Okay, let's pick on the males and talk about the male reproductive system. I should tell you right now that males, at least in terms of reproductive endocrinology, are boring. Okay? So the testis has basically two parts to it. The seminiferous tubules, where spermatogenesis occurs, and we'll discuss spermatogenesis in just a little bit. Also found are the Sertoli cells. The Sertoli cells are kind of the nurse cells for sperm, but also has some endocrine function, releasing inhibin. The second compartment are the interstitial cells, known as the Leydig cells. And Leydig cells are the main endocrine cells of the testis. They respond to LH and FSH interactions. They have LH and FSH receptors. which do two things. One, they can influence spermatogenesis, but more importantly, they activate testosterone release. Okay. And again, the gonadotropins and their effects on testosterone or inhibin release. So let's talk about meiosis in the male, also known as spermatogenesis. During testis development in utero, the male fetus, the growing testis surrounds the embryonic germ cells, those cells that are destined to become sperm. The germ cells develop into spermatogonia, which via mitosis increases in lots and lots of numbers, like a couple of million per testis. Again, still in utero, spermatogonia may also begin meiosis, which takes one of the spermatogonia and goes through that reduction division that we discussed way back. We talked about genetics to form spermatids eventually. Spermatids move to the seminiferous tubules, mature, grow a tail, and become sperm. Okay. Again, here is the spermatogenesis chart. Spermatogonia developed during fetal development, produce lots and lots of spermatogonia. The spermatogonia will continue to lie within the testis throughout the lifetime of the male. Thus, spermatogenesis is a lifelong event. They first form the primary spermatocyte, which undergoes the first meiotic division to form secondary spermatocytes. Each one of those then goes through the meiotic division once again to form spermatids, which mature to form sperm. Thus, one spermatocyte produces 4 sperm. Given that male sperm count is among the highest of mammals, there are a lot of spermatogonia still around within the testis. So again, males produce and deposit the most sperm per mil. But their males also produce a bunch of aberrant sperm. It is thought that up to 50% of the sperm delivery are aberrant. They're missing a tail, very common. They have 2 heads. The tails don't function. These aberrant sperm are seem to be caused by all sorts of environmental and chemical conditions, but they also may work as a sperm plug. Sociologists, anthropologists are studying the fact that it is possible during human evolution that males developed aberrant sperm count to block, shall we say, sneaky males from depositing their own. All right, we've gotten rid of the boring males. Let's discuss the much more interesting female reproductive system in terms of meiosis, the 28-day menstrual cycle, and of course, pregnancy. Okay, so again, there is the female gonads known as the ovaries. Ovaries are the site of oogenesis and the production of eggs, also as well as sex hormone and pregnancy hormone production. At the tip of the uterus are the fallopian tubes or the uterine tubes that suck in the egg that has just been released by the ovary during ovulation. The fallopian tubes are also the place where human fertilization normally occurs. Finally, there's the uterus. The uterus is going to serve as the development site for the attachment of the fertilized egg and the production of that unique mammalian invention known as the placenta. Again, here is the drawing of female parts, including the unusual association between the ovary and the uterine tubes. Okay, so within the ovary, these primary oocytes are within the periphery of each ovary. A couple of a month or so prior to their development, one primary follicle starts to produce and develop into what is known as the secondary follicle. In response to luteinizing hormone and follicle stimulating hormone, these follicles now start to develop a larger layer of surrounding cells known as the granulosa cells. These granulosa cells during the menstrual cycle will serve as the endocrine organ of the ovary. Okay? So again, one primary follicle is chosen. Normally, in rare cases, it may be multiple ones, and that's how you get fraternal twins, not identical twins. Okay, this is again showing you this nice light micrograph showing you the primary follicles on the surface. You can see there's a single cell layer that surrounds the primary oocyte. Again, one will develop into the secondary follicle, and in the middle is the secondary oocyte. All right, I still talk about oogenesis. If that primary follicle, the primary oocyte, is chosen to go through the next cycle, it immediately starts going through meiosis 1, the reduction division. It does produce 2 cells, but only one cell continues. That second cell degenerates as what is called a polar body. The secondary oocyte stays, gets poised and ready to begin meiosis 2, the secondary step. But that secondary oocyte will never complete meiosis 2 unless it is fertilized by sperm. This photo shows an enlargement of the secondary follicle. In the middle is the secondary oocyte. On the outer layer of the secondary follicle is a very thick layer of granulosa cells. Granulosa cells are the ones that are going to produce the major hormone pre-ovulation, the estrogens. Again, this is just showing you a picture of a secondary oocyte that has been pulled out of the secondary follicle. And you'll see the polar body, which was that second cell that was produced, but it degenerates. So let's look at oogenesis, shall we? Again, like spermatogonia, you have oogonia in the fetal female, and prior to birth, the oogonium via mitosis duplicates itself. We have a couple of million. Each of them are turned into a primary oocyte contained within the primary follicle. One gets chosen to begin the development of into the secondary oocyte, goes through the first meiotic division, but instead of two cells being produced like we saw with the sperm, only one cell is produced as that second cell degenerates. Now meiosis is arrested. The secondary oocyte, which is now being produced, goes through the process that we'll talk about in the next set of slides, which is the menstrual cycle, and gets ready for ovulation, or the removal of it from the follicle into the fallopian tube. If that secondary oocyte is invaded by a sneaky sperm and fertilization occurs, this will trigger the completion of that second meiotic division, where the secondary oocyte produces two cells. One is the fertilized egg that will immediately fuse its nucleus with the sperm's nucleus, and a second cell known as, again, polar body, which is removed and degenerates. Thus, unlike the primary spermatocytes, where one primary spermatocyte will eventually form 4 sperm, one primary oocyte will produce 1 egg. All right, take a deep breath as we will continue going through the slides of the uterine or menstrual cycle. All right. Actually, menstrual cycles, as done by humans and their related group, the primates, is rather rare in mammals because of the secretory phase of the buildup. So we can call this the uterine cycle because we can follow how the changes within the uterus. First, of course, is the loss of material via the menstrual cycle, followed by a estrogen and eventually progesterone effective activation of growth of the endometrium, known as the proliferative phase. And finally, if during the during after ovulation, there's the secretory phase, in which progesterone helps prepare the endometrium for attachment. And you can correlate the uterine phase with the ovarian phase. There is development of the follicles, ovulation, and then control of the possible pregnancy via the luteal phase. Luteal phase. All of this together makes up the human 28-day menstrual cycle. Okay, shall we go with it? All right, so menstrual cycles always begin with the first day of menses, the period. Okay, somewhere around day 7 or so, there is an increase in GnRH release, which causes an increase in FSH and LH release, which goes down to the follicle cells, stimulates the granulosa cells, remember those, to produce estrogens like estradiol. Here, estradiol does two things. One, it activates growth within the endometrial lining, right? The proliferative phase from the previous line. But estrogens also go back to the hypothalamus and the anterior pituitary and cause the release of more GnRH, LH, FSH, or at least does not have an effect on these glands. Thus, prior to ovulation, in this 5 to 7 day period, estrogens have a positive feedback effect on its release. This causes a very quick and large production of estrogens. So you can see this during the hormonal changes. You'll see LH and FSH, there's no GnRH here, but will also rise. Again, somewhere like 7 days before. This causes an increase in estrogen production, which can go back to the hypothalamus, anterior pituitaries, and have a positive or at least no feedback effect. Bottom line is, prior to ovulation, there is no negative feedback effects by estrogen. After ovulation, which would be on the next set of slides, there is a huge drop in LH and FSH production, and that is caused by the combination of estrogen, which resorts back to its negative feedback effects, and the pregnancy hormone known as progesterone. And we'll discuss that again in a few more slides. Ovulation. Ovulation is caused by that fast production of LH and FSH. Again, ovulation is caused by LH and FSH. This causes huge changes to the ovary, especially the secondary follicle, breaks the membrane, and the secondary oocyte is forced out into the abdominal cavity, very close to the fallopian or uterine tube, which using cilia brings the secondary oocyte into that structure. Kind of a neat microscopic view of the oocyte being released very close to the cilia of the uterine tube. Right, so we've had pre-ovulation, which causes a couple of things: getting the uterus ready for the endometrial lining of the uterus ready for implantation. Also causes, because of the surges of LH and FSH, ovulation. Now, post-ovulation, the ruptured follicle continues to become the endocrine gland, but it changes shape, it changes its color, and becomes a structure known as the corpus luteum. Again, that secondary follicle that has just released the secondary oocyte during ovulation alters its shape, alters its function, alters its color to become what is known as the corpus luteum. The corpus luteum is now the post-ovulation endocrine gland, and it secretes estrogens and the pregnancy hormone known as progesterone. Both estrogen and progesterone now begin to mature the endometrium for implantation of the fertilized egg. As was discussed in a slide before, the production of estrogens, the production of progesterone have strong negative feedback effects on the hypothalamus and the anterior pituitary, so that it inhibits GnRH, FSH, or LH release. Here is a photo of the corpus luteum post-ovulation. Okay, so still talking about the corpus luteum. The corpus luteum is producing lots and lots of estrogen and progesterone up until around day 25 of this 28-day cycle. If there is no attachment of a fertilized egg, either because it failed to attach or there was no fertilization, the corpus luteum begins to degenerate. And if you look back on the slide or upcoming slides, you'll see that estrogen and progesterone production drops. Progesterone, if you remember from a couple of lectures ago when we talked about RU486, one of the functions of progesterone and its cytoplasmic receptor within uterine smooth muscle is to block the production of prostaglandin, specifically prostaglandin E, by blocking the transcription of prostaglandin synthase genes. The increase in prostaglandin levels within the smooth muscle of the uterus causes smooth muscle contraction or menstruation. Here is a nice diagram of the ovarian cycle. starting with the primary follicle, primary oocyte, maturing into the secondary oocyte, ovulation, development of the corpus luteum, and then degeneration of the corpus luteum. During the proliferative phase within the uterus, the endometrium is being stimulated at first by estrogens, second after ovulation by estrogen and progesterone, to develop, to quickly go through mitotic changes where the endometrium increases in size. Correspondingly, there is increase in the development of capillaries to feed these growing endometrium. Again, preparing it for fertilized egg attachment. When the corpus luteum degenerates, it stops producing estrogen and progesterone, causing menstruation. All right, let's look at a nice chart, put everything together. Okay, on the top is the amount of the gonadotropins being produced. The second row is what's happening during ovarian development and follicle development. The third level are the steroid hormones involved, the sex hormone estrogen, and the pregnancy hormone progesterone. And the bottom one is showing you what's happening with the growth of the endometrium within the uterus. Again, I'll go through the menstrual cycle, the whole shebang. Okay. Somewhere around day 7, there is a rise in LH and FSH production. This causes a rise in estrogen production. Prior to ovulation, which usually occurs around day 14, estrogens have a positive or no effect on LH, FSH, GnRH production. At day 14, thanks to estrogen production, there is a surge in the amount of LH and FSH being produced. It is this surge of LH and FSH that causes ovulation. The secondary oocyte exits the follicle cells, exits the ovary. Immediately, the remaining follicle cells begin to change their shape, their size, their function, their color, and become the corpus luteum. The corpus luteum immediately starts producing progesterone and eventually, a day or two later, estrogen. The estrogen and progesterone have two basic functions: prepare the endometrium for attachment and block production of LH and FSH. Okay. Somewhere around day 25, if the corpus luteum has received no signals that the fertilized egg has attached to the uterus, it begins to degenerate. And you see this at day 25 as levels of progesterone, levels of estrogen begin to drop. Once these steroid hormone levels drop to a certain point, this triggers menstruation. Before we get into pregnancy, we need to talk about how to prevent pregnancy birth control pills. Birth control pills are usually a synthetic blend of estrogens and progesterone that do two things. One, the mixture, the blend of these two steroid hormones, which you can take orally, by the way, mimics the normal human 28-day menstrual cycle in terms of the uterine cycle, in terms of the buildup and then the release. Okay. It will also allow the secondary oocyte to begin its, and the follicle cells around it, its maturation processes. However, because of the addition of progesterone early on, this blend of steroid hormones produces not a positive feedback effect of GnRH, LH, and FSH, but a negative feedback effect, which means there is no surge of GnRH, LH, and FSH near by day 14, which means the secondary oocyte never leaves the ovary. Ovulation never occurs. So, if there is no secondary oocyte within the fallopian tubes to be fertilized by sperm, pregnancy will not take place. Okay. So again, the traditional common birth control pill are merely blends of estradiol and progesterone taken at certain days to mimic their normal peaks. However, with the addition of progesterone early on, this prevents the day 14 surge of LH and FSH, preventing ovulation. Thus, for a woman to be on a birth control pill, they have to take a pill every day. There are some pills, because you also want to mimic the drop in estrogen and progesterone effects, that have no hormones inside. They're placebos. Again, the purpose of this medication is twofold: to try to maintain the normal effects on the uterus, the endometrium, but also prevent ovulation. Oops, you forgot to take your pill. Fertilization has occurred, which means we're going to talk about pregnancy and giving birth or parturition. Again, males deposit among the highest amount of sperm of all mammals. Some of them may survive, and if the timing is right, the sperm will move into the egg nucleus and fertilization occurs. Fertilization actually occurs in two processes. The first process is that there's this calcium wave that occurs after the first sperm nucleus enters the egg cytoplasm. Actually, I should call it the secondary oocyte cytoplasm, because the egg hasn't fully developed yet. This post-fertilization calcium waves does two things. One, it stimulates the egg to produce a secondary fertilization membrane, which prevents other sperm from entering the egg. Secondly, this high level of calcium, right? It's always calcium. Activates the secondary oocyte to continue through meiosis to initially become the haploid egg, the final product. Again, this is just showing you what is occurring during the final steps of fertilization of the secondary oocyte. Right? You see on the right, fertilization occurs, the development of that calcium wave, preventing other sperm from getting in, and also activating meiosis 2, which causes the secondary oocyte to go through meiosis, releasing that second polar body, which will degenerate, allowing the sperm nucleus and the egg nucleus to fuse, producing the fertilized egg, you. Well, at least you some 20 some odd years ago. Again, here are some photos of human fertilized eggs and fertilized egg development as they move into all these divided cell stages. So let's talk about this fertilization process, at least in terms of stem cells. Okay, now if that early fertilized egg, the 2, 4, or 8, even 8 cell stage, become divided somehow, some way, those 2 divided sets, whether it's the 2 cell stage, most likely, occasionally the 4, rarely it's the 8, if those things are divided in half, those cells are still capable of dividing, capable of becoming blastocyst, capable of becoming identical twins. The other type of twinning, fraternal twins, are for 2 different fertilized eggs that were shed at that same time. Very rare, and it's basically like, you sharing the uterus with your brother or sister. So even though they're called twins, they are not the same as identical twins. Identical twins share the same genetic component. Okay? Now, human cloning. Human cloning can occur in 2 ways. One, identical twins, right? Clone of the same egg. The other way you can do it, well, has been proposed to do it, is to take a nucleus from any cell in your body. Okay? You then take that nucleus out and insert it into a recently fertilized egg in which you have removed that fertilized egg's nucleus. You put the other body cell, known as a somatic cell, into the cytoplasm of the egg, and you let the processes and the influences of that cytoplasm, plus a few drugs you add, convert that adult nucleus now into a fertilized egg nucleus. This is how lots and lots of animals have been created. Have you ever heard of Dolly the sheep? Produced, what, 30 some odd years ago now? She was produced by somatic cell nuclear transfer. Okay, so those are cloning. What about the embryonic stem cells? These stem cells are what is known as pluripotent, which means they can give rise to any type of cell in your body, but cannot themselves produce, be cloned into an organism, a whole organism. Okay, again, pluripotent. They can be altered into any cell in your body through magical ways, well, genetic and biochemical ways. Okay, and these pluripotent cells are found within the blastocyst. If you take a look at the previous slide or the next slide I'll show you, you'll see that there is this layer called the inner cell mass. Those are going to become you. Well, were you, or have become you. And they can give rise to every other cell, every other tissue layer in your body. If you take those cells out and you culture them with certain magical, genetic, chemical, and other biological features, they can become any cell in your body, which means you can use these cells for cell replacement. Perhaps within your pancreas to replace the dead beta cells to secrete diabetes. How about in your brain to secrete dopamine to cure you of Parkinson's disease? Or how about complete the transfer of signal in spinal cord injury? Okay, so again, after the blastocyst, on day 5 or so, the fertilized egg that you can see on the left, including that nice layer of the inner cell mass, attaches to the endometrium. The implantation of fertilized egg to the endometrial lining causes 2 things to happen. 1, there is this interesting interaction between embryonic tissue and maternal tissue, right? That's mom. These 2 cells kind of communicate chemically and begin the process of that unique mammalian invention known as the placenta. The second interaction that occurs during implantation of fertilized egg with maternal endometrial tissue is our last hormone, applause please, of the course, human chorionic gonadotropin. Within, some people think within hours of implantation, the endometrial cells get influenced and begin to produce this good-sized protein known as HCG, human chorionic gonadotropin. HCG has a couple of interesting functions. The first and primary function is it goes back to the ovary and keeps the corpus luteum alive, prevents it from degenerating, to allow the corpus luteum to continue to produce estrogen and that pregnancy hormone, progesterone. It does this for the next month or so until the placenta takes over to become the main endocrine organ of pregnancy, and placental cells produce estrogen and progesterone until the end. Okay? By keeping the corpus luteum alive, this prevents menses, keeps the endometrial cells nice and thick and vascularized. Okay, so here's a nice view of the amount of hormone produced, and you see this quick, very fast rise of HCG almost immediately. HCG is also famous for being that chemical that you look for on those $35 pregnancy tests that you can buy from CVS and Rite Aid and other stores. It is simply a antigen antibody reaction that these kits will turn color, looking for the presence of HCG in urine. Right? And so the drop that you put on has HCG antibodies on top of it. Okay? The best way to determine whether or not you are pregnant is not to buy those $35 tests. Instead, it is to go to a doctor and do an HCG blood test. They are much more reliable than the urine tests, because the urine tests, the antibodies aren't quite as good, and there's a lot of false positives or false negatives if the level of HCG isn't high enough in the urine. Now, I said there was going to be a second function of HCG. Please note that HCG does not actually get to zero, as the placenta continues to produce HCG in relatively low levels for the rest of pregnancy. And HCG only leaves the female only after birth. Sorry guys, you cannot produce HCG naturally. So what is the function of HCG at low levels? Well, HCG has some structural similarities to a hormone we previously talked about, thyroid stimulating hormone, or TSH. So possible function of this low level of HCG to augment TSH release, which increases the amount of thyroid hormones produced. Again, this is in mother, which is in the mom, which increases mom's metabolism. Right? Remember, mom is now feeding for two, and her metabolism needs to be much higher than when she wasn't pregnant. Okay? One more thing about HCG. Because of its mimic of TSH, Olympic athletes are now injecting themselves with HCG, both males and females. Okay, a few more slides. Let's again talk a little bit about that unique mammalian invention known as the placenta. So here's Junior now developing within the uterus, and he is interacting with mom via the placenta. The placenta, fetal and maternal blood never mix, but their capillaries get very, very close together to allow diffusion and osmosis to occur. So again, there is this very close interaction between mom and fetus. The blood does not, of the two individuals, do not mix. However, the capillaries get so close together that materials can move. So nutrients, oxygen, can go from mom to Junior, and Junior can give mom all his waste products and carbon dioxide. Again, just a slide showing the non-mixing of maternal and fetal blood. We discussed this before, but just before we give birth and end the class, remember that the placenta becomes an endocrine organ a few months into pregnancy, where it produces both estrogens, estradiol and estriol, and the pregnancy hormone progesterone. And remember that progesterone, via one of its influences, is to inhibit smooth muscle contraction of the uterus to maintain pregnancy, right? All right, we're ready to get rid of this class, get rid of this baby. So, what causes labor? Well, if you think about labor, and parturition is just the whole stage of childbirth, labor is the activation of uterine smooth muscles. Remember, smooth muscles are supposed to be weak, contracting? And we do know the hormone involved in the stimulation of those weak, smooth muscle contractions of the uterus. Oxytocin, right? We know oxytocin is the positive feedback and blah, 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 blah. That was a couple of lectures ago. Okay, so we need to start it. So, who do you think should make that decision? Mom or Junior? Correct. Remember Thanksgiving? This was so much easier in the fall semester because near Thanksgiving, right? And you'd put the turkey in, and you bought that turkey with the pop-up thermometer. Junior is the pop-up thermometer. Okay. The key here is fetal cortisol. Yes, Junior can make fetal cortisol. Junior makes fetal cortisol throughout development, but cortisol levels are usually quite low. During the last few weeks of pregnancy, fetal cortisol levels increase dramatically. And it accomplishes a couple of things. Number one, we need to do the final maturation of the last two organs that you didn't need during fetal life. The liver, right? There's no need to have a functioning liver, is there? Mom does all the breakdown. Mom gets rid of all your waste products and detoxifies things before she gives it to you. And secondly, of course, the lungs. You don't need the lungs to breathe in a liquid environment. Fetal cortisol triggers the final development of the liver. You need those cells so you can immediately break down fetal hemoglobin into adult hemoglobin, right? Remember bilirubin and its toxicity, especially in newborn babies. And development of the alveoli and drying out of the alveoli layer, which is filled with water in fetal stages. And this process takes about 2 weeks before actual childbirth begins. Okay, so cortisol stimulates the production of this, production of surfactant, drawing out of the liquid, maturation of liver cells. But fetal cortisol is high enough where it moves out of the fetus and into the uterine smooth muscles. It's a steroid, Nothing is going to block it from going in, especially in a liquid environment. And these levels of cortisol is high enough where it can get to the smooth muscle of the uterus. There, cortisol blocks progesterone from binding to its cytoplasmic receptors. Since the progesterone receptor complex is required to block prostaglandin synthase, transcription in uterine smooth muscles, now it cannot do that. Prostaglandin synthase can now start being made in smooth muscles. Prostaglandins cause smooth muscle contractions in the uterus. So smooth muscles now begin to contract. Thanks again to cortisol, fetal cortisol. Okay? These contractions are very irregular and are known as either Braxton Hicks contractions or the more common name, false labor. Some of these false labor contractions can be felt by mom, depending upon time, day, whatever. And what these false labor contractions are doing is pushing Junior, who has now changed so he's head down, towards the cervix. Remember the bottom part of the uterus. Junior starts beating his head against the cervix, activating those stretch receptors, causing the hypothalamus to produce and transfer oxytocin to the posterior pituitary, where oxytocin is released, binds to the smooth muscle contractions, and begin the rhythmical, strong contractions known as labor. We are done. Good luck with exam 4.